Dear students, in this class, let us know about the molecular basis of mutations. The term mutation was given by a person known as Devers in the year 1901. Now, what do you mean by mutations? Any sudden change occurring in the hereditary material is called as mutation. These mutations may be harmful, beneficial, or sometimes neutral. There are many ways as to how we can classify the mutations. Depending upon where they act upon, we can classify them as gene mutations. In gene mutations itself, we can see that there will be the mutation which results in the change in the phenotypic effect. That means to say, there might be change in the amino acid sequence. When there is change in the amino acid sequence, this amino acid change might bring about change in the phenotype of a particular character. Otherwise, we have basically in the types of mutations, we have basically two types of mutations, somatic mutation and germline mutation. So what are somatic mutations? These are the mutations which occurs in the somatic cells. These mutations, when happen in somatic cells, they are passed on to the other cells through the process of mitosis. The effect of these mutations depends on the type of cell in which they occur and the developmental stage of the organism. If they occur early in development, larger the clone of the mutated cells. The next type of mutation is germline mutation. These are the mutations which occur in the cells known as gametic cells. And this type of mutations are passed on to future generations. In multicellular organisms, usually the term mutation is used for germline mutations. Depending upon how the mutations occur, we mainly have two types of mutation. One is spontaneous, the other one is induced mutations. When we speak about induced mutations, it is mainly happened by an external agent where we call them as mutagens. These mutagens might be physical mutagens or chemical mutagens. Under chemical mutagens, we have many chemicals which might bring about the change in the sequence of the DNA and thereby change the sequence of the protein. And the chemical mutagens are alkylating agents, base analogs, methylating agents, DNA intercalating agents, DNA cross-linking agents, and reactive oxygen species. We also have the mutagens, which comes under physical agents, where we call them as UV radiation and ionizing radiation. This slide depicts the structure of different type of chemical agents. Examples being alkylating agents. In alkylating agents, we have ethyl methane sulfonate, we have ethyl ethane sulfonate. We also have base analogs, acridins, deaminating agents, and hydroxylating agents. As told before, we have basically two types of mutations depending upon how they occur, and the one being spontaneous mutations. So, what are spontaneous mutations? These are the mutations which are happening because of the natural changes. Mostly, spontaneous mutations causes depurination and deamination. What do you mean by depurination? Removal of a purine base from a DNA molecule. Deamination, removal of an amino group which is present in the nitrogenous base of a DNA molecule. Otherwise, as told, we have induced mutations. These are the mutations which are induced by the environmental agents, usually the agents being the physical agents and the chemical agents. Any environmental agent that increases the rate of mutation above the spontaneous is called as a mutagen, and they are usually physical and chemical mutagens. As told, spontaneous mutation is mainly because of deamination. Now, here, this particular slide gives an example of deamination where we have a nitrogenous base cytosine, and the molecular name of this nitrogenous base is 2-oxy-4-aminopyrimidine. This might undergo deamination, meaning to say removal of amino group. And that removal of amino group takes place in the fourth position of the base, wherein deamination of cytosine gave rise to uracil. Now, uracil being a regular base, a normal base, but it is exclusively present in the RNA molecule. It so happens sometimes the mutations are gone unnoticed. The next type of mutation, example, what we are seeing here is of a modified nitrogenous base, and that we call it as 5-methyl cytosine. This 5-methyl cytosine on deamination give rise to a regular base known as thymine. Thymine, once again, being a nitrogenous base of a DNA molecule, mainly indicates that the mutation has not happened. But these are the main important reason for the cancerous type of cells wherein even though the mutation has happened, the proofreading mechanism does not recognize them as the wrong base because they are the regular bases. We can dwell on this particular topic in the later classes. You also have an example of deamination, which happens because of the chemicals, and the chemicals usually being nitrous acid. Now, this particular slide speaks about the deamination of the nitrogenous bases such as adenine, cytosine, and guanine. Adenine, on reaction with nitrous acid, gets converted to a compound known as hypoxanthin because there is a removal of amino base. Now, this hypoxanthin behaves as guanine, and thereby they are going to get paired with another nitrogenous base where we call them as cytosine. As a result, adenine thymine base pair gets converted to guanine cytosine, which is an example of transition mutation. The next example is of another nitrogenous base, a pyrimidine cytosine. Cytosine, as I told you before, on deamination gives rise to uracil, a regular base, where this uracil now attracts the adenine nitrogenous base, where now it seems to be as a regular base pair, meaning to think that there is no mutation. But however, mutation has happened. The third example is of guanine. Guanine, on deamination, they give rise to a particular non-modified base where we call them as xanthine. Xanthine still base pairs with cytosine, but the difference here is that in guanine cytosine we have three hydrogen bonds, but whereas here we see that xanthine and cytosine is base paired with two nitrogenous bases, meaning to say that the connection or the bond between them might be weak because the number of hydrogen bonds are just two hydrogen bonds. Now, when we try to speak about how the mutation happens when we have a deamination happening on a nitrogenous base known as cytosine. Now, towards the right, it's a particular double-stranded DNA, where in the double-stranded DNA, we see that there was cytosine, and this cytosine has been deaminated. Since it is deaminated, now we see that in the second position, you have uracil. So what happens during DNA replication? This double-stranded DNA splits into two single strands where each of them act as a template, and as a result, the strand which is carrying the uracil, a modified base, now it will try to give rise to a new strand wherein you will be having the second base pair as UA base pair. Actually, in the parent strand, it had to be CG. CG is now getting converted to UA in the strand where you have a modified base known as uracil. The other template, it is a normal template where it is giving rise to a double-stranded DNA wherein they are not changed, meaning to say they are the same as that of the parent strand. But now considering the mutated strand, when this mutated strand, now what they do is that they are indicated that the DNA has undergone mutation. Even though uracil is a regular base, the mutation has happened. Next to this particular figure, we have another example where you have a double-stranded DNA. In the double-stranded DNA, purine is lost. Which purine is lost? Adenine is lost. So as a result, it so happens when there is a loss of base, loss of a nitrogenous base. In this particular example, it so happens that particular that DNA which is trying to enter into the DNA replication, the DNA with the strand which is having a gap, they just fold upon. But whereas the other strand which is having an unpaired base. They have what they have an unpaired base, wherein what they do is that to the unpaired base, the correct base might come and form a hydrogen bond. So as a result, in a double-stranded DNA, if you have a strand having depurinated one will be mutated, the other will be unchanged.